So good morning in Boston and the US. Good evening in India. Good afternoon in the UK and Europe. And in Japan, I guess it's good night. I know we have visitors from Japan as well. So welcome to season 2 of ChaiTime. Through popular demand, we came back with another 5 episodes that we sort of pre-planned, covering areas within the biotech therapeutic ecosystem, whether it's infrastructure or clusters and science topics. And we've had some very broad range of interesting topics with a focus on investing as well in the space. And today I've got my very good friend, Jennifer Petter on the virtual couch. We will have Ros with us shortly. We're just trying to find out where she is, but Jennifer is a very well-known person within the biotech sector, and it's a real honor to have her on my virtual couch today. So welcome, Jennifer.
So Neil, you should have me as well, but maybe you can't see me.
I couldn't see you. You're here. Brilliant. Okay.
Sorry, I shouldn't set up investor calls straight before I'm due on a panel.
Well, welcome, Ros. So I can now welcome my second guest today to the ChaiTime. So we have Ros here, who's CEO of o2h, and are you in Oxford at the moment?
Just south of Oxford, about half an hour south of Oxford.
You're in Oxford, and Jennifer, I guess, is in Waltham, I guess?
Well, no, actually, I'm in the lovely town of Stowe, Massachusetts, which is just outside Boston. So,
yeah, welcome to you both. It's a real honor to have you both on as guests on our ChaiTime. And I think we have a session that will last around 45 minutes. And first of all, I would like you to both introduce yourselves and give our viewers a little bit about your background. So how about Jennifer first?
Okay, I'm a chemist by training, Dartmouth, Duke, Columbia, and organic chemistry to be specific. I had a disreputable academic career for a while at the University of Pittsburgh, and then moved to industry very happily in 1991. Spent 5 years at Sandoz, which of course you now know is as Novartis, and then moved to Biogen in 1996, where I ended up heading up small molecule drug discovery. Left there in 2005 with 650 of my best friends. Spent about 1.5 years at Mersana Therapeutics running research, and then about another 5 years at Avila Therapeutics running drug discovery. And then we got bought by Celgene, which of course now is inside of BMS, but at the time got bought by Celgene, and ended up being VP of chemistry for Celgene for a few years. And then left there in 2015 to found Arrakis Therapeutics, which is where I work now.
Thank you very much. So Ros, maybe you'd like to introduce yourself and give us a bit of your background.
Yes, I'm Ros Deegan. I'm currently CEO of o2h Therapeutics, which is an Oxford-based company leveraging native mass spectrometry for drug discovery in high definition. My career started as a scientist, but after a series of accidents, including when I tried for work experience at a vet, suffice it to say that I passed out before the dog, I ended up in industry. And that was really just a homecoming for me. It was the first time that I sort of felt like I could leverage my skills from a broad perspective, and the fact that I'm not a good practical scientist while still being embedded in the discipline I love of science. And so I was much more comfortable on the business side. I went through a number of roles in management consulting, then went and did an MBA. And then I joined business development, and I'm a kind of real generalist. And so business development, I think, is the only sort of function where generalists can survive, because every deal is a little bit of everything. It's a little bit of research. It's a little bit of commercial. It's a little bit of finance, a little bit of sort of organizational people stuff. And it's, quite a lot of legal as well. So I did that for a few years at GSK. And then I had my first biotech experience back in 2008. I joined a company called Trevena. At this point, I was in the US, so I spent the last 13 years in the US, not too far away from Jennifer. I used to live in Concord and was working in Lexington. My first biotech, I was there for 7 years. I headed up business development, finance, and operations, a company called Trevena. helped take that from Series A through B, C, and then public, and progressed our programs from, first imaginings to clinical assets. And then I went and led the US subsidiary for UK company, Bicycle Therapeutics, and had a sort of accelerated experience there where, again, went through the sort of private public, or at least when I left, we just confidentially filed to go public, and also that transition from preclinical to clinical. And I moved back to the UK to join OMAS, not actually purposely. I was looking for companies that had a European connection, but I wasn't thinking I was going to relocate. And then I fell in love with the company, and it was only after I'd fallen in love with the company that I sort of sat myself down and said, well, hang on, Ros, there are 20 employees and they're all in Oxford. Do you really think you're going to be a Boston-based CEO? So I decided to move myself and my family to Oxford.
I thought you were about to say when you realised the company was based in Oxford and I studied in Cambridge.
Well, that as well. I mean,
what am I doing in Oxford?
I'm a complete Oxford advocate now, though. So.
All right, okay, you're converted. We'll come back to a little bit more on your background and some of the things that you've been up to in a second. I'm going to spotlight Jennifer first, and I'm going to address the obvious elephant in the room in a way. So I knew Jennifer when Jennifer was known as Ros, and I've known Jennifer and Ros before for probably around 18, 20 years. And when the news came out that Jennifer was now transgender, I was like, yeah, that's okay, fine. I just, it's just one of those things that it didn't really affect me. But it is a big deal, and obviously it's a big deal for Jennifer. And I've followed the news. Michael Gilman put out an amazing blog. Who is the CEO of Arrakis and who was leading that at the time, and a close friend of Jennifer's. And it must have been a very difficult time. So I'm just going to really have some open questions around Jennifer, given what's going on in the world. With everything, everyone is really more aware around equality, Black Lives Matter campaigns, and this area is nothing different. So the biotech industry has traditionally been very, very conservative. The new generation, I think the younger kids just really brush it off and don't, when I talk to my children about this area, they're like, big deal, Dad, what's the big deal? So, but I just wondered, so to Jennifer, the first question is, when you decided at the age of 60 plus, I won't go into the exact age if I don't quite know it, that you decided to come out as transgender, why did you decide now was the right time? And why did you not decide earlier? I think that's a good question to start with.
First of all, just to talk about the kids, yeah, so this particular child, who's now 17, when I was 15 at the time that I explained the situation to her, and yeah, she kind of brushed it off, sort of like, okay, does this mean we get to go shopping? To which I said, yes, it does. Let me just say, why now as opposed to earlier? I will say that, in the 60s and 70s, there was no vocabulary for this. There was no way for us to find each other. There were frankly, very few cases of people who transitioned. So it just plain looked impossible. And so you just remove it from your life and you get on with things, and you have families and children. And I would say, perhaps the real wake-up call for me was when Kathy got triple negative breast cancer. That turned out brilliantly, all right, but it was kind of a rough ride, and it shakes you, and it makes you think about who you are and where you're going, and I think that in ways that were not entirely cognitive, it really sort of changed the direction of my life. So you begin to move in that direction. In fact, and oddly, you begin to move in that direction before you entirely understand that you are moving in that direction. But it became something I explicitly began to plan around. starting around, I'd say, mid-2016.
And how has the industry reacted to you coming out as transgender?
Well, here's the thing. So I think you're right. There is a certain sort of conservatism, because I think that may partly be fueled by scientists wanting to see a certain purity to their science, and to keep personal and social and political issues at arm's length. But the reality is that what I found was absolutely everyone wanted to seem like the most woke person in the room, right? Everyone wanted to seem just so chill about everything. So the result is that you end up living in this bubble. So I've lived a very privileged life, and there are still elements of that sort of bubble that have followed me down this path. So I certainly had anxieties that this would go poorly, and let's just say had to develop at least mental contingencies to how I would deal with that. But people have been fantastic about this, and so I mean, just to maybe the way, a very material way to express that is that I came out publicly in May of 2018. And then in that summer, I went full time, as we say, in June of 18, and spent the summer and fall on the campaign for our Series B. And no one blinked an eyelash.
Well, you've raised shed a load of money, so it can't be that bad for you.
Yeah.
Yeah, I mean, you came to visit our facilities in India, and I do remember, having, we had that fire, we had the dancing, and trying to get you to dance, and so we had some really fond memories of you as Ros, and hopefully we can have some more further fond memories as you as Jennifer. So, I mean, you've had some great success in your career. I met you when you were at Masana, actually, and you then moved on to Avila. Tell me a little bit about Avila, and why do you think Avila was particularly successful? It's a very different play to, it was more of a chemistry play and a sort of a warhead play rather than a platform biology play where you are today. But why do you think Avila was particularly successful? What did you learn from your experience there? You weren't the founder of Avila, you joined the team, didn't you?
No, I was employee 10. So, and at Mersana, I was employee 4. So actually, let me just say, I do see a through line between these, and that in all 3 cases, Mersana and Avila Therapeutics and at Arrakis, the fundamental problem that needs to be solved is a molecular one. So at Mersana, you say, well, we don't like PEG, so we need something better that decomposes and is highly soluble. And so that was what they called Fleximer. And then you look for problems you can solve with that. Similarly, actually, at Avila Therapeutics, what you do is you look at sort of known kinases and proteases and ask, if we could make this covalent, how would that improve the pharmacology? And so you go through, in essence, a structural bioinformatics exercise to look for those opportunities, find molecules that you can turn into covalent inhibitors, and then see if you can, in fact, solve some kind of pharmacology problem or toxicology problem. And then, so I mean, I think that in that case, again, although the biology follows the chemistry, to a large extent, that is also the case at Arrakis Therapeutics, where you look at RNA, folded RNA, and you say, can't we drug this with small molecules? And having now found many molecules that bind to these RNAs, you then begin to look for where are the kinds of biology problems that we can solve, having solved the chemistry problem. So, so to me, there's a through line of molecularity driving those programs.
I mean, thinking back to my sort of biochemistry days, trying to drug RNA just seems like, I think you're one of the first companies to attempt this. I know there are 1 or 2 others now. But how do you even find a drug target that can help, reshape or refold protein? How do you even find a target on an RNA, and then how do you validate that target? I think that's probably the most. I think with the advances in chemistry, we can find ways of drugging stuff drugging the undruggable. And I know that, that's a particular expertise of yours, but how do you validate those targets? I think that's the thing I can't get my head around.
So that turns out to be like the bulk of what we do, is to correctly identify, first of all, reasonably stable folded structures in the messenger RNA, and then to make sure that those structures are in some way confluent with a mechanism that we think is interesting, whether that's ribosomal docking, translation, splicing, degradation. And then the effort is to say, well, can we, when you synthesize that segment of the RNA and demonstrate that it's well folded, will small molecules bind to it? And so we do screening for binding. It's a binding format to find molecules. And then the challenge is, to swim upstream a bit and find, is all well and good that the molecules bind, but do they have impact on the sort of career of that RNA and the biology inside the cells? So, I'm not sure I can provide you with a lot of detail without a couple hours and a CDA.
I thought you were just going to throw out a target in there that we could go around such a drug it. But yeah, I'm just going to dip down to Ros for a few minutes. So Ros, you're actually a rower from what I read, and you're a captain of the female rowing team.
I was vice captain at one point.
So you're one of those people that shout at people to get out of bed at 4:30 in the morning. I would get out of bed if you shouted at me.
Exactly. I think someone actually shouted at me. I've been single sculling recently, which is interesting because normally I rowed kind of 8s. And once you got used to rowing, an 8 is incredibly stable. And the only rowing we can do at my rowing club right now is single sculling. And it's absolutely terrifying because the boats are so unstable. So I fell in the Thames last week.
Well, I've been swimming in the Cam, where you used to, your old haunt in a way. Recovered from the ear infection, which I had a few weeks ago from that. But coming back to your career in a way, so it looks like you spent a significant amount of your learning and your time at GSK and running that virtual drug discovery unit, and that was probably sort of the early 2000s, and that's when sort of the virtual drug discovery thing was really just coming into play. Did you sort of pioneer that, and what kind of deals did you do when you were part of that group?
Yeah, I mean, it was very much coming in at that point, I think, to let the biotech companies have more control of the process in the deal, and to also do deals which weren't sort of straight asset licenses, but were collaborative, and where the pharma partner was sort of providing guidance but not direction. These sort of option-based deals. So I'm not sure if I pioneered it. I was on the original deal that GSK did with Exelixis, actually before I was part of the virtual discovery group, which I think was one of the first of that kind, but there were a series being done at that point by certain companies. But I think actually dedicated resource to just doing that at GSK was quite novel, because we had all the functions represented. We had BD internal to that group. So, the kinds of deals we did were typically option-based deals. We did a deal with, we actually bought, I think it was called it's a while ago now, that was called Praxis, that was really interesting. Was it called, or Plexagen? I can't even remember now. We did a deal with Galapagos, but then we, which I think included, the JAK inhibitor, but then maybe didn't option it. We did a deal with Chemocentrics. We did a deal with Ranbaxy in India under that time period. And I led a deal with Oncomed at the time in cancer stem cells. So it was a very productive period of really bringing in a lot of different innovation. And part of the deal was that we would sort of slightly counter to it didn't matter if the rest of the organization didn't buy into it. It was designed to be a little bit to encourage counter thinking.
When I look at your career, all the different industry, and I guess that's what attracted the investors of OMAS to bring you into that, because I can see that that's a platform that you're going to have to do a huge amount of deal making to develop that company. But I mean, you've done, I was trying to count it up, but I lost count after like $1 billion. It's a lot. You've been part of a lot of deals, and so it's a real congratulations to you on that. Not very many people have actually really done those things. But coming to your sort of time in OMAS, are you are you there to sort of really now commercialize that platform and do deals with the pharma companies?
It's interesting.
Maybe you could just give us a little bit. I mean, I know a little bit about OMAS, obviously, but maybe you can tell people listening a little bit about OMAS, the amount of funding that you've raised and so far, and what your strategy is.
Yeah. So we are, as I said, native mass spectrometry. So our founding scientist is a fantastic lady called Carol Robinson, who was actually the first professor of chemistry at Cambridge University, and then also the first professor of Cambridge at Oxford University in chemistry. And she has really spent decades pioneering, or at least over a decade, pioneering the use of native mass spectrometry, or mass spectrometry as a tool to actually interrogate protein complex biology. So to actually leverage within a single screen the binding and the function of the target through mass change. And if you think about GPCRs, which is one of the areas that we're looking at initially, everything a GPCR does is a function of mass change. When a ligand binds, changes mass. When it couples with a G protein, changes mass. When it couples with a different G protein or with beta-arrestin, that's a different mass change. And you can actually pick all that up in a very highly purified system that you can manipulate multiplex. So it's a really cool platform technology. In terms of the deal aspect, I mean, I introduced myself as a generalist rather than the BD person, and that is because that's always sort of been my gig, really. Although I had a strong transactional theme to my career, I wasn't exclusively BD. So even in the Bicycle role, I was leading and setting up the US subsidiary in addition to BD. In Trevena, I was head of finance operations and BD. And so it's interesting, you say the assumption being that I must be coming in to partner that platform. Our investors are Syncona and OSI, and we raised over 40 million in capital. And the model is actually to own our own assets and to take them to market. So that doesn't mean that we won't partner, but it's not a partnering model. It's, I think because it's a platform, there will be ways to generate value through partnership. And we're very targeted in terms of where we think partnering makes sense and where it doesn't. But we're not, we're in a very privileged position with the capital that we've raised and the backing that we have. And the programs that we've now initiated within our portfolio, that we're not under any pressure to partner. Okay. And for me, I actually, I feel much more passionate about the quality of a deal than the quantity of deals.
Yeah. I think that, as you evolve in your career, you kind of sense that, okay, I want to make, if I do a deal, it's got to be significant now. It's at least got to be bigger than the last one I did, kind of thing. But Jennifer, in Arrakis, you've done obviously a huge amount of fundraising and very successfully. And I think we had this, I remember having a coffee with you, and you were saying, I know we were trying to raise money, we were trying to plot how to do it. And you were like, when you brought Mike Gilman into the company, when he joined the company, I think you told me that if he decided to set up a laundromat in Boston, everyone would be like, oh yeah, that's a great business idea, we should do that. He had that magic about him. So how's the fundraising gone in Arrakis, and tell us about the recent deal that you've done.
So the first fundraising was remarkably easy. It was one breakfast with Raj Parekh, and we got seed funding, about 2 million, and actually Henry Termeer participated as a co-founder, and that was really wonderful to be part of like his last couple of years. And we got to work generating some data, and you were involved at that stage as well. The A round, we started to look around, and Mike had already joined on the board, and he decided in the fall of 2016 to join as CEO at the same time he became CEO of Obsidian. And that was catalytic to give people comfort about us, leading to the A round of about 38. And I should say, in terms of answering your question about how that was, on the one hand, you present to a lot of people, and a lot of people were very skeptical about the platform companies more generally, but specifically the challenges of drugging RNA. But you find a few believers, and they appreciate the magnitude of the opportunity. And then so then we moved on, generated a much broader set of data, particularly solving the screening problem. And that culminated in a Series B of 75. So the Series A was Advent Life Sciences and Henry Termeer and Pfizer and Celgene, Oh cripes, I'm having a blank. Canaan Partners, who actually led the round. And then the B round came in, it was all those people plus Nextech and Venbio co-led it, and Omega and Google Ventures, and I think a couple parties I'm forgetting, comprise the B round. And then, as you may know, earlier this year, we finally did our first transaction with a pharma company. And that was fairly substantial. It was, the upfront was 190 with a gazillion dollars in biobucks in the background that we'll see if we can activate those. And again, I will say that raising the B had its challenges, but the transactional opportunity that was very different. It was like being the prettiest girl at the dance. There was a lot of interest in what the company had to offer, and so we're delighted with the partner.
Congratulations on that, Jennifer. That is an amazing feat to raise that finance and convince people to invest in groundbreaking science and an evolving area, as in the RNA, trying to drug RNA, and then being able to do a colossal deal with the big pharma like that. That's brilliant news. And it kind of evolves me into the next part of my, which is what I really wanted to ask, is I've spent a lot of my career, as has Ros, kind of going between the US and the UK, and I've seen things done differently in Massachusetts and San Francisco. I've sort of been selling chemistry service for the last 15 years, and I'm sort of evolving to other things as well as doing that as well, as we all know. But I've seen a lot of how drug discovery is done in the US and how we do it in the UK, and I'm always trying to make comparisons. And I've written down for sort of 5 or 6 points here. I'm going to like blast them at you, and then you can comment. So one is chemical space. I think in the US, once they decide they want to drug a target, they really drug the target. They take out the space so well, and it's partly due to the funding environment, which is part of the questioning really. And in the UK, we sometimes, not always, go a little softer and leave ourselves more holes. The valuations in the US are significantly higher in the US than in the UK. At the seed round, surprisingly, they're a bit higher, but as soon as it moves into Series A and B, they do, I think, multitude up. But even at the seed round, they're probably 2 or 3x different, probably. I've put here, throw the kitchen sink. I think in Boston, they throw the kitchen sink at it and just go all out, and they find a plan B and C very quickly. In the UK, we have unbelievably brilliant science, but sometimes we don't throw the kitchen sink at an area, even if we are leading in the area. And why and what is, I may have got this wrong, but I'd be interested to hear your opinion. I think in Boston, the scientists have very, very good business acumen. Almost they're grown up with the commercial thinking, whereas our scientists have some of that, but they are really out and out scientists. And that's maybe part of the education system, that we just focus on, a small number of areas at a degree level. And then the other thing I wrote, which I'm not quite sure how to apply it, but it's a science first approach, not finance first. And it's having a good grounding on the science first. And I think we've actually quite good at that in the UK as well, but even more so in the US. And you can see that when you approach investors in the US, they grill you on the science in a way that you may not get the same level of grilling in the UK, may not. Some of the more progressive VCs are very different on that respect. Because it's a more mature industry at the seed stage, you definitely get that more in the US and the UK. In the UK, you may get non-scientific investors who like the idea of a particular therapeutic area and having an impact in that space, but may not have the scientific people around them to do the necessary due diligence. So these are some of the things I just thought I'm probably going to get lots of messages around after that from various things I sit on. But what are your thoughts on that? Did I get that completely wrong? And how can we evolve that discussion? So I want to go to Ros first on this one, because she's bridged both in a way that I found very interesting, which is why I thought,
yeah, I mean, I think you know, a lot of your points kind of come back to investor in the end, because I think that sort of throwing the kitchen sink, you can't do that if you don't have capital. Narrow chemistry, can't do that if you don't have capital. And some of them come down to sort of cultural differences. And I think that both are true. I mean, I've been in a very privileged position with OMAS because Syncona is very US-like in their approach, and o2h as well. And so, you know, I would not in any way say that we don't get a scientific grilling. You know, I'd like to see a scientific grilling if we don't have one. So they're very diligent, and so as a company we're very diligent, and we're also very well backed. So we also, you know, do things properly, and we have the ability to do it properly. And success breeds success as well, because you attract very high quality individuals to companies such as OMAS. So, you know, like my CSO came from Heptares, where he was chief technology officer, and Heptares was a very successful membrane protein GPCR focused company. We also have I had a chemistry from Heptares. You know, head of pharmacology comes from Novartis, and again is a very experienced GPCR pharmacologist. So we have incredible talent at OMAS, but that's because we are sort of a big fish in a small pond, and so we've been able to attract these very, very high quality individuals, but they're not everywhere. And so that's the trouble, is that if you're not sort of one of the sort of golden few, then it'd be quite hard to also get that talent. And it's not that, and it's talent that has it's the experience and that sort of business acumen. And I hadn't thought of it as kind of education based. I just felt that I feel that my 13 years in the US gave me an ability to sort of experience, you know, biotech on a fast track that I would really have struggled to achieve in the UK, because you don't have all those companies going through all of those, you know, really high quality capital raises, high quality science, taking the, you know, listing. You know, Bicycle, I think, was the first Cambridge UK company to biotech to list on NASDAQ, and, you know, and Cambridge is one of the hotspots of biotech in the US, in the UK. And so it's really hard to find people who've just been there, done that. And so even though they're very smart people and scientifically they're very smart, they don't have that experience, which is also so important to sort of think about the consequences of how you're thinking.
I should carry on. I think we have got the experience, but not as much as they have it in the US. They've got it everywhere. We have to really, but the numbers are a lot smaller. I think that's the problem we have in the UK.
Yeah, there's fewer people. And so at OMAS, I feel very good about the quality, but I think recruitment is more challenging. It's more challenging in Boston because everybody leaves, because, you know, people leave before they've even signed their offer letter. You know, you kind of like sign it and they come for a day and then they've left or something. So you know, the other thing that I think culturally is maybe slightly different, and in some ways I understand US culture better than British culture, because I was there for 13 years and I've only been back for one. But I still think there's a little bit of a pharma biotech disconnect between the two countries. I think in Boston, it almost got to the stage where being in pharma was just so kind of boring. You know, it's like it defines you as you're embarrassed when you go to a dinner party and you're kind of like, oh, I'm at a big company, and everybody in biotech is cool. Whereas I still think in the UK, it's not the same. I still think there's a sense of, you know, being at GSK or AstraZeneca is a cooler thing than being at a really hot biotech company. And therefore, sometimes it's harder to...
Well, how do you find how do you find the valuation discussion? Because it really puzzles me. In the UK, we work on intellectual property for exactly the same market. We're not making drugs just for the NHS. We're making drugs for Europe, Japan, US, and all the other markets, and filing the same patents. Our scientists.
We're losing you, Sunil.
Why the... Right, can you hear me now? I don't know.
Yes.
Okay.
Oh, he's breaking up. So I think you're asking about valuation. So,
I'm asking about evaluation. I'll close some stuff down while I do that. Maybe I've got too much open.
So I'll talk about, yeah, I mean, absolutely, there is genuinely a valuation discount, but I think it comes back to, you know, the fact that the capital is harder to get in the UK. So of course, you know, there's a discount applied in order to get capital. But I think that hopefully as and I do think we're at an earlier stage in the growth curve, and we're in a good place. I think, Oxford has, I think, demonstrated even during, the last few months of the COVID pandemic, the technology and the strength of the technology in Oxford. You've got, one of the leading vaccine candidates. We've got one of the sort of most, interesting sort of fast test approaches is coming out of Oxford. And then also the trial infrastructure in the UK is very strong. So, the data on the recovery trial, I think, shows the robust clinical trial network in the UK. So there's definitely pieces that are very strong. And I think we're in a good place on the growth curve. But I think for, the discounts are going to continue for a while until there's a surplus of sort of capital, as opposed to where there isn't at the moment. So there's a discount.
Can you hear me now, by the way?
You break up a little bit.
Are you able to hear me now?
Yeah.
Okay, right, okay. It's probably my kids on the playing Fortnite or something, taking up the bandwidth. But I was saying that basically we're targeting the same markets with our intellectual property. I think our scientists on both sides of the pond are equally as good, which is why it always confuses me around the valuation disconnect. And I'm always thinking, how can we bridge our biotechs into the US? And how can we make the ecosystem work together better? So Jennifer, maybe you have an opinion on some of the bullet points I threw out earlier, whether on how to make, where you think the strong points are in UK biotech and compare that to Boston.
So, I probably not qualified to make general comments about biotech or venture funding in the UK. But I will say that, but it's worth noting that our founder and seed company was Advent Life Sciences in London, and so it really it's a certain visionary element to their approach to take on this issue really so early, and invest fairly heavily for them in our mission. So. And they've really stuck with us the whole time. And of course, we got to know them first off when we also invested in Avila Therapeutics. So I will say that if there's something I don't know if this is being reproduced in England or the UK more generally, but a lot of the venture firms in the Boston area they don't simply have a waiting room and then bring people in like auditions. They actually see the companies themselves. I mean, the extreme expression of this is the Third Rock model, and then you see Atlas following a similar kind of approach to many companies. Flagship is getting in that direction. So I think that's something that contributes.
Do you think we're in a valuation bubble? I've seen, and I saw Bruce Booth's blog in Forbes this week, and he highlighted the amount of capital that's coming into our sector. And it's, given that what's happening in the world, it's kind of the opposite to what you expect. But I think that my opinion is that generalist investors want to invest in biotech. They're generally underweight the sector, and they want to allocate some money to what they see as groundbreaking science and how to do that. So I think we're going to see a lot more money coming into our sector. Do we think that will lead to a valuation bubble?
Well,
a crash. What are you seeing in terms of valuations out there?
So we haven't really gone through that process, and so I'm not actually, I don't have the data that you're looking for. I will speculate that what's appealing about us is that we look like the solution to the problems. We also have, it's also more of a long-term game, so that you're not looking at companies that actually have to reach customers through a supply chain at this point, and so that makes us fairly attractive. So, but as far as the valuations go, well, all I could say is that certainly the IPOs are a fairly sort of high-profile way to look at that, and those are going extremely well.
Yep.
I think it's going to depend a bit, isn't it? So it's going to depend how sort of all of this plays out. But I do worry a little bit, because I think that although we're not immediately affected, sort of, health continues to be a need, more so than ever. in terms of actual our ability to progress science, there are limitations on sort of clinical trials right now. And so, you know, will we be able to get everything that we were working on to the same point that we would have done without the pandemic? You know, probably not. I think it's been pretty impressive how much we are able to continue, but there will be delays in things, and that therefore should impact value. And we're not seeing that.
Are you seeing further digitization of our sector, particularly? I mean, I'm thinking when you mentioned clinical trials, if we can people can be more remote, collect data more remotely, and have control over processes to do that digitally. Are you seeing technologies emerging that maybe will speed up the overall drug discovery and development processes? And we can actually flick to, you know, how you see AI impacting our industry as well as it impacts every industry. So to Ros, maybe first.
Yeah, I mean, I think as a discipline, it's certainly becoming more important, and there are companies that are very focused in that area. And so I think it's watch this space right now. It's too early to sort of, you know, the proof is in the pudding. So when we actually kind of get these molecules that have been, but there are molecules being developed quicker with more of an AI bent to their drug discovery process, and if they become good molecules, but it's such a long process that we don't get to test the hypothesis except for over like a 10-year period. Yep. And it's too early. We haven't tested the hypothesis yet, but one would expect that it will. It's just how much.
And Jennifer?
Yeah, I mean, we're just so deeply into the discovery phase that many of those issues as they pertain to clinical data and sort of feedback, you know, pharmacovigilance, all of that's so far downstream for us. I would say that, you know, for us, big data is really about, you know, terabyte level next-gen screening. Next-gen sequencing, excuse me.
Right, yeah. And where do you see are the next frontiers in sort of our sector? Where you've looked at sort of the RNA space. Where next for what's coming in, the most exciting thing that you can think of?
Well, it's an interesting question. No, so I think the protein degraders are doing well. There may be some variations on that. It may not be completely played out. I actually am seeing some interesting science about the prospects for getting proteins inside cells. And if that becomes a practical option, that could be quite revolutionary.
Yep. And Ros? Where next for our
it's
industry?
funny because my head of chemistry just teams me to say that we just did a 477 million compound virtual high throughput screen in 24 hours. So I don't know if we're going to have any good compounds that come out of it, but I do think, you know, those things could be quite transformative.
Yeah.
So I think I mean, clearly I'm passionate about the ability of our platform to change how people think about drug discovery. I think the biophysical approaches give a really a higher resolution, higher content readout. I think there's a lot we don't know that we still think we know about protein biology. And the interesting thing for our platform is even when we look at really well-studied receptors as our kind of exemplars, we still find out new things by leveraging mass spectrometry because it's such a detailed, high content readout. And so I think this is, you know, going to be transformative to how people think about drug discovery and will be widely applied beyond OMAS. And it's already starting to be. I think that, you know, that another area that we're particularly interested in is some of the, you know, drug classes that are less well explored. So while we've started with GPCRs, we're also looking at solute carriers as an area that is very untapped and where it's difficult to do cell-based assays and where we think there could be some really exciting new biology and new targets that we can apply our platform to uniquely. So that's a new frontier for us.
I've got a long list of questions. I would love to just sit and chat with you both more. But we're at the end of our time. So unless there's anything else that you think that you wanted to add, I would like to thank you both for your time today. I think it's been really interesting discussion, and I'll hopefully catch up with you after this call. So thank you to both of you. Our next ChaiTime is in 2 weeks' time, when we have Big Brother Shah on the show, and he will be chairing a panel looking at architecture and shape. So we have two fairly large infrastructure projects ongoing, which he's leading those teams. And he'll be looking at how shapes and architecture can shape culture. So that should be quite an interesting discussion. A couple of weeks from now, emails will follow. Thank you very much, everybody, and we'll see you soon.
Thank you very much. Enjoyed it.
Cheers. Thank you so much.